The overall goal of this procedure is to perform injection of cells, dye, or DNA in embryonic mouse hearts in order to investigate cell fate or to locally manipulate gene expression. This is accomplished by first dissecting out the embryos from a pregnant mouse and exposing the embryonic heart. Next, DNA, dye, or cells are injected into the targeted region of interest in the heart. Then, after the embryo is incubated, the heart is removed and placed in culture. Finally, the fate of injected or labeled cells is determined via microscopy. Ultimately, the fate of injected, labeled, or genetically modified cells is monitored by confocal microscopy or light and epifluorescence microscopy, revealing the functional importance of specific genes and cells during cardiac development.
The main advantage of this technique over existing methods like transgenesis in mouse is that we can locally and accurately inject, label, or genetically modify cells in vivo. This allows for the investigation of these cells in the proper environment.
Demonstrating this procedure will be me and Emilye Hiriart, a PhD student from my laboratory.
After preparing equipment, reagents, and solutions according to the text protocol,